fancy term for the formation of blood clots is hemostasis. And we need to have hemostasis occur so we do not bleed out because it prevents blood loss. You need to know these 3 steps in order. The very first thing is that you will have a vascular spasm, and that basically just means that the smooth muscle in the vessel wall is going to contract to help limit the size of the damage. A platelet plug will then form. Platelets will release a hormone, which is known as serotonin, which can be a neurotransmitter, depending on what cell releases it. That's hormone, serotonin, will tell the blood vessel to vasoconstrict even more, or the wall of the blood vessel to vasoconstrict even more. And then you will have coagulation, which is the actual formation of a blood clot. So let's go over these again, a little bit more info. Very first step, vascular spasm. The blood vessel wall will immediately vasoconstrict. Step 2, platelet plug formation. When that endothelial wall is damaged, platelets will start to become sort of sticky and sort of spiky, and that means they'll adhere to each other and the damage in the wall vessel. Once they're attached, other platelets are going to be pulled into and attracted to the site of injury. And that's actually a positive feedback mechanism in order to help with clot formation. And then you have coagulation. This is a multi-step process, and it's way more complex than we go over. Basically what happens is, in order to get blood to clot, it is transformed from a liquid into a gel. There are clotting factors or procoagulants which promote clotting. And there are also going to be those that are going to inhibit clot formation. Those guys are going to be called anticoagulants. There are 3 phases to the clotting process. The first is the formation of a prothrombin activator. Then you will convert prothrombin to the active form of thrombin. And in addition to that, you will form a fibrin mesh from fibrinogen in plasma. So what happens after that? The clot will retract. And basically that's a process during which the contractile proteins that are within those platelets are going to contract and pull on the neighboring fibrin strands. They will actually squeeze plasma from the clot and pull the edges of the tissue closer together. There will be a factor known as platelet-derived growth factor that will stimulate repair. Fibrinolysis. You can probably tell what that is if you know what the parts of that word mean. Means that fibrinolysis means that unneeded clots are going to be removed because of an enzyme, plasmin, that is going to digest fibrin. Great video on that, so make sure that you watch that. Now, what limits clot growth or clot formation? There are 2 mechanisms that are going to do that. Blood is going to be moving past the area, and that will basically disseminate or spread clotting factors before they can actually trigger a clotting cascade. Or activated clotting factors are going to be inhibited. The active form, thrombin, that isn't bound up to fibrin is going to be inactivated by a factor which is known as antithrombin 3, as well as a protein called protein C, and heparin. Remember heparin? As long as the wall of the blood vessel, that vascular endothelium, is nice and smooth and intact, platelets are prevented from clotting because they cannot attach to that lovely smooth wall.